My name is Caitlin Candell. Today is December 9th, 2025, and this is my cell bio final presentation.
My name is Caitlin Candell, and today I will be presenting my final Diseases of the Cell project on Wilson's disease, a rare but serious genetic disorder of copper metabolism. This presentation summarizes the cellular basis, genetic etiology, disease mechanisms, diagnosis, treatments, and emerging research on Wilson's disease. Wilson's disease is an autosomal recessive disorder caused by mutations in the ATP7B gene, which is responsible for removing excess copper from the liver and incorporating it into ceruloplasmin. When ATP7B is defective, copper can no longer be excreted properly, so it begins accumulating in the liver and eventually spreads to the brain, eyes, kidneys, and other organs. This accumulation leads to progressive tissue toxicity and organ damage. Though rare, early detection is critical because the treatment can prevent irreversible neurological or hepatic decline. The ATP7B gene plays a central role in copper homeostasis. Under normal conditions, ATP7B is located in the trans-Golgi network of hepatocytes. Its job is to load copper onto ceruloplasmin, the major copper-carrying protein, and to excrete excess copper into bile. This prevents copper from building up to toxic levels. ATP7B also indirectly supports mitochondrial function, redox balance, and the activity of many copper-dependent enzymes. When this gene is mutated, all these processes are disrupted, leading to cellular stress and toxicity. Wilson's disease is caused by mutations in ATP7B on chromosome 13q14.3. Over 900 different mutations have been identified, including missense, nonsense, insertion deletion, and splice site variants. One of the most common is H1069Q. This is found especially in European populations. The disease follows an autosomal recessive inheritance pattern, meaning affected individuals must inherit two mutated alleles. Carriers who have only one mutated allele are typically asymptomatic. If both parents carry the affected allele, each child has a 25% chance of being affected, a 50% chance of being just a carrier, and a 25% chance of inheriting two normal alleles. Genetic testing and carrier screening are extremely valuable tools for at-risk families. When ATP7B is dysfunctional, copper accumulates inside cells, especially hepatocytes, and causes several harmful effects. Excess copper interferes with mitochondrial oxidative phosphorylation, reducing activity of complexes 2, 3, and 4. This lowers ATP production and increases reactive oxygen species, or ROS. The buildup of copper also triggers regulated forms of cell death, including cuproctosis, a copper-dependent mechanism that disrupts mitochondrial proteins. Additionally, impaired ceruloplasmin production contributes to abnormal iron metabolism. Together, these cellular disruptions lead to tissue injury and organ dysfunction. Wilson's disease affects diverse organ systems due to toxic copper accumulation. The liver serves as the principal site of deposition, often resulting in hepatitis-like presentations, cirrhosis, or acute hepatic failure. Neurological involvement, particularly of the basal ganglia, engenders movement disorders, which includes tremors, dystonia, and psychiatric symptoms. Ocular copper deposits in the cornea produce distinctive Kaiser-Fleischer rings. Renal and hematologic systems are also impacted, causing hemolysis and renal tubular dysfunction. Musculoskeletal and endocrine complications can include osteoporosis, arthropathy, and hormonal abnormalities. The multisystem disease impairs critical physiological functions. Metabolic regulations are compromised, adversely affecting energy production, detoxification of reactive oxygen species, and metal homeostasis. Neurological and motor capabilities, including coordination, cognition, and mood, are diminished. Hepatic processes such as detoxification, excretion, bile regulation, and the metabolism of drugs and proteins are all disrupted. Hematological and renal capacities, including erythrocyte integrity and waste filtration, are impaired. And the musculoskeletal health suffers through reduced bone mineral density and joint dysfunction. Collectively, these disturbances result in global disruption of homeostasis and multi-organ impairment. Diagnosing Wilson's disease requires a combination of clinical evaluation and specialized tests. Patients often undergo serum ceruloplasmin testing, which is typically found to be low, and a 24-hour urinary copper test, which is typically found to be elevated. A slit lamp eye exam may reveal Kayser-Fleischer rings. In uncertain cases, a liver biopsy can directly measure copper levels. Genetic testing for ATP7B mutations provides confirmation and is strongly recommended for symptomatic individuals and family members of those who have been diagnosed with Wilson's disease. Because early diagnosis prevents progression, clinical awareness is extremely important. Several effective treatments exist to manage Wilson's disease, and most patients can live a normal life if they begin therapy early. The main treatment approach is chelating agents, such as D-penicillamine or trientine, which bind to copper and help eliminate it through the urine. Zinc therapy is another widely used method. Zinc blocks intestinal absorption of copper and is often used for maintenance or pre-symptomatic patients. Dietary changes, such as limiting high copper foods, are also recommended. In severe cases, particularly when liver failure occurs, liver transplantation is a curative solution because the new liver contains a functioning ATP7B gene. Recent research on Wilson's disease is increasingly focused on gene therapy, especially through adeno-associated viral vectors to deliver a functional ATP7B gene directly to liver cells. Animal studies have shown promising improvements in copper metabolism. Researchers are investigating the role of epigenetics as DNA methylation patterns may influence disease severity and onset. Biotechnology has produced preventative options for families with a known history of the disease. Genetic carrier testing allows prospective parents to determine whether they carry a mutation in the ATP7B gene, and this information can guide reproductive planning. Pre-implantation genetic diagnosis, used in combination with in vitro fertilization, enables the selection of embryos that do not carry mutations associated with Wilson's disease. These technologies empower families to make informed decisions and reduce the likelihood of passing the disease in future generations trials. Apoptosis, a newly described cell death pathway triggered by copper, is an emerging therapeutic target. These developments highlight how rapidly biotechnology is advancing our understanding of this disease. At this time, there is no complete cure for Wilson's disease, and patients require lifelong copper management. However, the outlook has improved dramatically over the past few decades. Gene therapy offers the strongest potential for a one-time, long-lasting treatment that may restore ATP7B function. As molecular research continues to clarify copper-induced cell death pathways, targeted drug therapies may also become possible. With continued advances in biotechnology, a definitive cure is becoming increasingly realistic for future generations. To conclude, Wilson's disease is a powerful example of how a single gene mutation can disrupt fundamental cellular processes and lead to widespread organ dysfunction. Understanding the role of ATP7B and copper metabolism provides important insights into the disease's etiology, systems, and treatment. While current therapies are effective at preventing progression, cutting-edge research, including gene therapy and epigenetic investigations, offers promising hope for improved outcomes and potentially curative treatments. Thank you for listening to my presentation. Have a good night.